Hello there. Let's begin our journey into neural simulations using Neuron with Python. First, we need to import the Neuron simulation environment and Matplotlib for plotting our graphs. Then we need to create the SOMA. So to create a SOMA or a cell body, we type in SOMA equals age, that section, our section, and we will call it SOMA. So to make our SOMA spherical, we set its length and diameter to be the same value. Here we set them to be 20. In order to proceed with our simulations, we need a model or a mechanism. So here we are going to add the Hodgkin-Huxley mechanism, HH. This mechanism models ion channels that allow action potentials. We do that by typing soma.insert HH. So in this video, we are going to be testing the response of a neuron to different light intensities. So we are going to set a list of light intensities for different values. Then we are going to create a list for voltage traces. So these light intensities represent, in real biology, would affect photoreceptors. But here in our modeling, we are going to use different current amplitude. So instead of light intensities, we are going to inject different current uh, values into our cell body or into our soma. This would have the same effect of different light intensities. So when we inject the current, we need to uh, record the voltage of our membrane over time, of course. So to do that, for each intensity, as we have 4 here, we are going to use a for loop. So for each intensity, we are going to inject a current, record the neuron's membrane voltage, and save the result for later plotting. And of course, these results will be saved in this list voltage traces. Okay, great. Now let's start with the for loop. Explain each part of it. For intensity in light intensities. I'm sure this is very obvious. Intensity is each value of these values that are present in the list. So for each one, we are going to do the same thing. So we are going to set stimulus to be h that IClamp. So this h that IClamp means that we are going to inject a current. This clamp word, you must have recognized it from the patch clamp technique. Then when we are going to where we are going to do the stimulus, it's in the soma, and we set the value to be 0.5, indicating that we are going to be doing that in the middle of the soma. Okay, then we are going to set the delay of the stimulus to be 5 milliseconds. This is a value just to visualize the graph. This is the delay that we will going to wait before injecting the current. Of course, for injecting a current, we can inject it for different time periods or different periods. So here we are going to set the duration of the stimulus to be 2 milliseconds. Then we set the stimulus amplitude. A stimulus amplitude would simply be intensity, but we have 4 of them, and for each one we are going to do the same thing using the for loop, of course. After that, after injecting the current, we are going to record the voltage using vec equals h dot vector dot record, from where we are going to record again from the soma, the middle of the soma. What are we going to record? The voltage, and to record the voltage we use point reference voltage v corresponding to voltage. And of course, for each recording, we would need to have a timing value also. So we are going to record the time also. Okay, great. And we record the time. Then we finalize by resetting our membrane potential to minus 65 millivolts for the next iteration of our list of light intensities, and we continue this run to 25 milliseconds. So we'll be having a graph that represents values of voltage from 0 to 25 milliseconds. That's all for the first for loop. So when we record these values of voltage and time, we need to store them. Where are we going to store them? In the voltage traces list that we have created before. So we type in voltage traces dot append intensity list of the T vec and list of the V vec. So for intensity is a single value, but for the time and the voltage, they are different values. These lists of T and V are what we are going to use to plot our different graphs. Okay, then we create a plot figure and we define its size to be 10 and 6. Then we trace our plots using a for loop again. So for intensity T, V in voltage traces, these values are present in the voltage traces list that we have created and filled using the for loop that we have just saw. For each one, we are going to plot the graph using the T values and the V values. And we are going to label each graph or each curve intensity, and we set that intensity to the corresponding intensity. So we have different intensities, and we will be having different graphs, 4 graphs. Then we going to just do some final plot tweaks, but before that we are going to set this is just optional. We are going to create a dashed line, a horizontal dashed line that would correspond to the typical firing threshold of neurons. We are going to set it to be minus 55 millivolts. Then we set the final plot tweaks. We label our axes, we give it a title, legend, grid, etc. Then we end up by showing our plot. So if I run this code, we will get a graph that represents the sensory neuron response to light intensities, different light intensities. As we see here, the first and second intensity are very low, so they do not give rise to action potential. But starting from 0.05 nanoamperes, we see that we get action potentials. So if you compare these action potentials, the one that we got from the intensity 0.05 and 0.1, they are pretty similar, but the action potential that we got from injecting a 0.1 nanoamperes current is very quick, so there is no delay here. It is not delayed like the one that we got from 0.05 nanoamperes. So we see that there is a difference here in the graphs, and that is very logical, and that's what we will get typically in the real biological cells. Different intensities give action potentials, but the difference is in the velocity or the frequency of the action potential. So here we see that the bigger or the higher the intensity, the quicker we get action potentials, and that is, as I said, very logical and aligns with what we see in real biological neurons. So for this simulation, we can change a lot of things. We can change the light intensities to see what we will get. We can change the duration of this stimuli, like we can run the simulation for 5 seconds and we see what we will get. We can change a lot of things. So as you might see here, when we change the the time or the duration of the stimulus, we get we get three action potentials. Even for the small intensity 0.04, we got an action potential. So this is another thing that interferes with the action potentials, or the cells given rise to action potentials or not. There is the intensity, there is the duration of the stimulus, and a lot of things.